day everyone, I'm Robert Jacob Yekyek and I'll be reporting oxidative phosphorylation. So, what is oxidative phosphorylation? It is the biochemical process by which ATP is synthesized from ADP using the energy derived from electron transfer and proton movement. Key players are electron donors, NADH, nicotinamide adenine dinucleotide, and FADH2, flavin adenine dinucleotide. Then the electron acceptor is the O2. Then outcome is ATP production. Significance: central to energy production in cells. Oxidative phosphorylation is the final stage of cellular respiration, where the energy from the breakdown of nutrients is used to produce adenosine triphosphate, or ATP, the primary energy currency of cells. It occurs in the mitochondria of eukaryotic cells and the cell membrane of prokaryotic cells. Concept of coupled reactions: pair of reactions where energy released by one is utilized by another. In oxidative phosphorylation, electron transfer in ETC is coupled with ATP synthesis, involves proton gradients and energy transduction. A coupled reaction is a pair of reactions. One is exergonic, releases energy, and one is endergonic, which absorbs energy, linked together so that the energy released by the exergonic reaction drives the endergonic reaction. The electron transport chain, or ETC, is a series of protein complexes and other molecules embedded in the inner mitochondrial membrane. So key components of ATC, complex 1, 2, 3, and 4 are electron transfer proteins. Cytochrome C and ubiquinone are mobile carriers. Functions of complex 1, 3, and 4 transfer electrons and act as proton pumps. So how does it work? Electron transfer are high-energy electrons carried by NADH and FADH2 from earlier stages of the cellular respiration are transferred to ETC. These electrons are passed along a series of protein complexes, undergoing a series of redox reactions. In each redox reaction, an electron is transferred from one molecule to another, releasing energy. Proton pumping in the ETC. Proton movement across the internal membrane. Complex 1, 4 protons. Complex 3, also 4 protons. And complex 4 has only 2 protons. Source of protons: reduced carriers NADH and FADH2, mitochondrial matrix. As electrons move through the protein complex in the ETC, energy is released. This energy is harnessed to actively transport protons from the mitochondrial matrix to the intramembrane space. This creates an electrochemical gradient across the inner mitochondrial membrane, with higher concentrations of proton in the intermembrane space. Chemisorter coupling is the process that links the electron transport chain to the synthesis of ATP through ATP synthase. It is a fundamental mechanism in cellular respiration and photosynthesis. Explanation of ATP synthesis driven by proton gradient, creation of electrochemical proton gradient, then followed by the flow of protons back into the matrix through ATP synthase, synthesis of ATP using energy from the gradient. Electrochemical proton gradient. An electrochemical proton gradient is a form of stored energy created by a difference in both concentration of protons and the electrical charge across a membrane. Components of the gradient: chemical gradient, high proton concentration in the intermembrane space versus matrix. Electrical gradient, positive charge in the intramembrane space versus the matrix. Roles of ATP synthase. Structure of ATP synthase. F0 subunit is the proton channel, and F1 subunit is the ATP synthesis site. Function of ATP synthesis: harnessing proton motive force. ATP synthase utilizes the potential energy stored in the proton gradient to drive the synthesis of ATP. Catalysis of phosphorylation. The enzyme catalyzes the phosphorylation of ADP to ATP, coupling this reaction to the flow of proton through the enzyme, maintaining proton gradient. Under certain conditions, ATP synthase can function in reverse, hydrolyzing ATP to pump protons against their electrochemical gradient. This helps maintain the proton gradient, which is essential for various cellular processes. Cellular energy balance. By coupling ATP synthesis to proton gradient, ATP synthase ensures the energy production is tightly linked to cellular needs. So this is the summary of oxidative phosphorylation. ETC generates proton gradient. ATP synthase converts proton motive force into ATP. Then significances, essential for cellular energy supply and highly efficient energy conversion mechanism.
ATP production for the common metabolic pathway. So first, let us recall what is metabolism. Metabolism is the sum of all biochemical reactions in a living organism. There are two main types of metabolism. First, we have catabolism. Catabolism is the set of metabolic pathways that breaks down molecules into smaller units that are either oxidized to release energy or used in other anabolic reactions. The second is anabolism. Anabolism is the set of metabolic pathways that construct molecules from smaller units. These reactions require energy, known also as an endergonic process. So the metabolic pathway is a sequence of biochemical reactions. It converts a starting material into an end product through step-by-step reactions. Each step is catalyzed by a specific enzyme to ensure efficiency in regulation. The common metabolic pathway. The common metabolic pathway refers to the final stages of energy production shared by carbohydrates, fats, and proteins. We have three different common metabolic pathways. First, we have the citric acid cycle, which generates NADH, FADH2, and GTP from acetyl CoA or acetyl coenzyme A. Second, electron transport chain. It uses NADH and FADH2 to produce ATP via oxidative phosphorylation. Lastly, we have the oxidative phosphorylation. It converts stored energy into ATP, the cell's energy currency. So now we're going to talk about the ATP production from the citric acid cycle. For each mole of NADH or nicotinamide adenine dinucleotide oxidized in the ETC, 2.5 moles of ATP are formed. FADH2, which stands for flavin adenine dinucleotide, which does not enter the ETC at its start, produces only 1.5 moles of ATP per mole of FADH2 oxidized. FADH2's entrance point into the chain, complex 2, is beyond the first proton pumping site, complex 1. Hence, fewer ATP molecules are produced from FADH2 than from NADH. Therefore, the yield energy in terms of ATP production can now be totaled for the common metabolic pathway. Every acetyl coenzyme A entering the citric acid cycle produces 3 NADH, 1 FADH2, and 1 GTP. Thus, 10 molecules of ATP are produced for each acetyl coenzyme catabolized. Now we have the figure or the cycle of the citric acid. So this figure on the left side relates the preceding 10 ATP production figures to individual steps in the citric acid cycle in which NADH, FADH2, and GTP are produced. Citric acid cycle origins for the ATP produced through operation of the electron transfer chain and oxidative phosphorylation per 1-acetyl coenzyme entering the citric acid cycle. Moving on to the next subtopic, we have the importance of ATP. The importance of ATP is that ATP is the link between energy production and energy use in cells. The conversion of ATP to ADP powers life processes, while the conversion of ADP back to ATP regenerates the energy expended during cellular operations. Lastly, ATP plays a central role in processes like muscle contraction, biosynthesis, active transport of nutrients. In the next slide, I will be talking about the importance of ATP. So in this structure, we can see the cycle of ATP to ADP cycling. ATP to ADP cycling is good for the energy exchange in cells. The cycling of ATP and ADP in metabolic processes is the primary medium for energy exchange in cells. This cycle allows cells to efficiently convert energy from food and other sources into a usable form to power life processes. Next, we have the ATP to ADP plus PI or inorganic phosphate powering life processes. The conversion of ATP to ADP plus the inorganic phosphate includes the biosynthesis of essential compounds, muscle contraction, and nutrient transport. As we all know, ATP is the energy source that fuels many essential cellular activities. ADP plus inorganic phosphate to ATP is regenerating energy. The conversion of inorganic phosphate plus ADP to ATP occurs in food catabolism cycles like glycolysis and the citric acid cycle. This regenerates the ATP that was expended in cellular processes, ensuring a continuous supply of energy. Here we have the figure that shows the interconversion of ATP and ADP as the principal medium for energy exchange in biochemical processes. This process is central to maintaining cellular function and energy flow and metabolism. ATP is the ideal source of energy for biochemical processes. Why? Because ATP is a high-energy phosphate compound that releases energy when hydrolyzed to ADP and inorganic phosphates. The hydrolysis of ATP releases an intermediate amount of energy. We have the 27.5 kilocalorie per mole, which is higher than the energy needed by some compounds that ATP donates energy to, but lower than the energy required to regenerate ATP. So why is this important? The energy from ATP hydrolysis is optimally suited for cellular processes. Like, it's enough to power these processes, but not too excessive to prevent the conversion of ADP back to ATP. If the hydrolysis energy were too high, the body would not be able to regenerate ATP because the energy required to synthesize ATP would exceed what is available. So now we have the key points. ATP is the energy currency of the cell driving essential biochemical processes. 2. Through pathways like oxidative phosphorylation, ATP is produced efficiently. 3. ATP powers critical functions such as muscle contraction, biosynthesis, and nutrient transport. 4th, the cycling between ATP and ADP maintains the energy balance within cells. Lastly, understanding ATP production highlights its role in sustaining health and life at the cellular level.
Good morning, everyone. Today we will explore a fascinating topic in biochemistry, non-electron transport chain oxygen-consuming reactions. While the electron transport chain in mitochondria is often at the forefront when we discuss oxygen utilization in cells, it is not the sole pathway where oxygen plays a crucial role. Cells engage in several alternative pathways to consume oxygen, with diverse purposes ranging from detoxification and biosynthesis to signaling and defense. Understanding these reactions not only deepens our appreciation of cellular complexity, but also sheds light on their implications in health, disease, and biotechnological applications. The electron transport chain, or the oxidative phosphorylation phase of metabolism, consumes more than 90% of the oxygen taken into the human body via respiration. As a normal part of metabolic chemistry, significant amounts of this remaining oxygen are converted into several highly reactive oxygen species, or ROS. Among these ROS are hydrogen peroxide, superoxide ion, and hydroxyl radical. The latter two of these substances are free radicals, substances that contain an impaired electron. Roles of ROS. Beneficial. It destroys invading bacteria or viruses and cellular signaling. It is harmful that it damages lipids, proteins, and DNA when unregulated. Sources of ROS. Internal, normal metabolism, and external, polluted air, cigarette smoke, and radiation. In the first step, superoxide ion is converted to hydrogen peroxide, which is in the second step converted into water and oxygen. Immediate destruction of the hydrogen peroxide produced in the first of these two steps is critical because if it persisted, then unwanted production of hydroxyl radical would occur via hydrogen peroxide's reaction with superoxide ion. Hydroxyl radicals quickly react with other substances by taking an electron from them. Such action usually causes bond breaking. Lipids in cell membranes are particularly vulnerable to such attack by hydroxyl radicals. It is estimated that 5% of the ROS escape destruction through normal channels. Operating within a cell is a backup system, a network of antioxidants to deal with this problem. Participating in this antioxidant network are glutathione, which is a cellular redox regulator. Another is vitamin C and E. These protect biomolecules. And lastly, beta-carotene, which is a lipid-soluble antioxidant, as well as other compounds obtained from plants through dietary intake. The vitamin antioxidants as well as the other antioxidants prevent oxidative damage by reacting with the harmful ROS oxidizing agents before they can react with other biologically important substances. Sources are diet rich in fruits, vegetables, and plant-based foods. Numerous studies indicate that diets high in fruits and vegetables are associated with a healthy lifestyle. One reason for this is that fruits and vegetables contain compounds called phytochemicals. Phytochemicals are compounds found in plants that have biochemical activity in the human body, even though they have no nutritional value. The functions that phytochemicals perform in the human body include antioxidant activity, cancer inhibition, cholesterol regulation, and anti-inflammatory properties. Each fruit and vegetable is a unique package of phytochemicals, so consuming a wide variety of fruits and vegetables provides the body with the broadest spectrum of benefits. In such a situation, many phytochemicals are consumed in small amounts. This approach is much safer than taking supplemental doses of particular phytochemicals. In larger doses, some phytochemicals are toxic. It can be toxic in large doses because they may interfere with cellular functions, cause oxidative stress, or disrupt metabolic pathways. Some act as natural defense compounds in plants, which at high concentrations can harm human cells, leading to toxicity. A major group of phytochemicals are the flavonoids, of which more than 4,000 individual compounds are known. All flavonoids are antioxidants, but some are stronger antioxidants than others, depending on their molecular structure. About 50 flavonoids are present in foods and in beverages derived from plants. Of particular importance as antioxidants in foods are flavonoids, known as flavones and flavonols. Flavonoids, whose core structures are enhanced by the presence of ketone and/or hydroxyl groups and a double bond in the oxygen-containing ring system. The formation of flavone and flavonol compounds normally depends on the action of light, so the highest concentration of these compounds generally occurs in leaves or in the skins of fruits, whereas only traces are found in parts of plants that grow below the ground. The common onion is, however, a well-known exception to this generalization. Quercetin, the most widespread flavonoid in foods. It is predominant in fruits, vegetables, and the leaves of various vegetables. In fruits, apple contain the highest amounts of quercetin, the majority of it being found in the outer tissues or of its skin. A small peeled apple contains about 5.7 milligrams of the antioxidant vitamin C. But the same amount of apple with the skin contains flavonoids and other phytochemicals that have the effect of 1,500 mg of vitamin C. Onions are also major dietary sources of quercetin. In addition to their antioxidant benefits, flavonoids may also help fight bacterial infections. Recent studies indicate that flavonoids can stop the growth of some strains of drug-resistant bacteria. Next, we will delve into a fascinating and essential topic that connects nutrition, biochemistry, and physiology: B vitamins and their role in the common metabolic pathway. These water-soluble vitamins are critical in maintaining optimal metabolic functions, serving as coenzymes that facilitate vital biochemical reactions. Metabolism, the site of life-sustaining chemical transformations within cells, depends heavily on these vitamins to extract energy from macronutrients like carbohydrates, fats, and proteins. Without their contribution, the pathways we rely on for energy production, such as glycolysis, the citric acid cycle, and oxidative phosphorylation, would falter. Structurally modified B vitamins function as coenzymes in metabolic pathways. Now that the reactions of the common metabolic pathway has been considered, it is useful to formalize in a summary fashion B vitamin involvement in the citric acid cycle and the electron transport chain. These are the 4 vitamins that have involvement in these metabolic reactions. First, niacin, second, riboflavin, third, thiamine, and fourth, pantothenic acid. Let's discuss more about these four vitamins. First, niacin, also known as vitamin B3, is a precursor to the coenzyme nicotinamide adenine dinucleotide. Once converted into NAD+ and is reduced from NADH, it plays a pivotal role in metabolism by acting as an electron carrier in energy-producing pathways. In glycolysis, its role, NAD+ is reduced to NADH during glycolysis when glyceraldehyde 3-phosphate is oxidized. This step is critical for maintaining the flow of glycolysis, generating ATP and providing NADH for further energy production. In citric acid cycle, NAD+ is reduced to NADH in multiple steps. For example, during the conversion of isocitrate to alpha ketoglutarate and malate to oxaloacetate. The NADH produced here is loaded with high-energy electrons, ready to fuel the electron transport chain. And lastly, electron transport chain. NADH donates electrons to complex 1 of the ETC, becoming oxidized back to NAD+. These electrons travel through the chain, ultimately reducing oxygen to water. The energy released during electron transfer is used to pump protons, the mitochondrial membrane, creating a gradient that powers ATP synthesis. Next is riboflavin. Riboflavin, or vitamin B2, is converted into two key derivatives, flavin mononucleotide and flavin adenine dinucleotide. These derivatives act as redox coenzymes, meaning they play a crucial role in oxidation reduction reactions, particularly in energy metabolism. Both FMN and FAD are involved in electron transfer, similar to NAD+ and NADH, but with different enzymatic functions. FAD is central in succinate dehydrogenase reaction in the citric acid cycle, or CAC. FAD is directly involved in the succinate dehydrogenase enzyme, which catalyzes the conversion of succinate to fumarate in the citric acid cycle, or the Krebs cycle. During this reaction, FAD is reduced to FADH2 as it accepts 2 electrons and 2 protons from succinate. Its significance: the FADH2 generated here carries high-energy electrons, which are crucial for ATP production in the electron transport chain. And second, electron donation in the electron transport chain, or ETC. Its role is that FADH2 produced in the succinate dehydrogenase reaction donates its electrons to complex 2 of the electron transport chain. From here, the electrons pass through the chain and contribute to the creation of a proton gradient, ultimately leading to ATP synthesis. Its significance is FADH2 contributes to AT production, but it generates slightly less energy compared to NADH. Each FADH2 molecule typically produces about 1.5 ATPs in the ETC, as it enters the chain at a lower energy level than NADH. Third is thiamine in decarboxylation reactions. Thiamine, or vitamin B1, is converted into its active form, thiamine pyrophosphate, or TPP, after being phosphorylated into the body. TPP is a crucial coenzyme involved in various biochemical reactions, particularly in carbohydrate metabolism. Its essential roles of TPP in metabolism is, first, pyruvate dehydrogenase complex, or PDC. Its role is the pyruvate dehydrogenase complex catalyzes the conversion of pyruvate from glycolysis into acetyl CoA, which enters the citric acid cycle, or Krebs cycle, for further energy production. TPP is required for the enzyme pyruvate dehydrogenase within this complex. Its mechanism: TPP acts as a coenzyme for the decarboxylation of pyruvate, helping to remove a carbon atom from pyruvate, transforming it into acetyl CoA. This reaction is a key step in linking glycolysis to the citric acid cycle. Its significance: the production of acetyl CoA is crucial for cellular energy production and the continuation of the citric acid cycle, which generates ATP. Second is alpha ketoglutarate dehydrogenase in the citric acid cycle, or CAC. Its role in the citric acid cycle, alpha ketoglutarate dehydrogenase, catalyzes the conversion of alpha ketoglutarate to succinyl CoA. This reaction, like the pyruvate dehydrogenase reaction, involves a decarboxylation step and TPP is needed for this enzymatic function. Its mechanism: TPP facilitates the decarboxylation of alpha-ketoglutarate, removing a carbon as CO2 and forming succinyl CoA, which continues through the citric acid cycle. The reaction is vital for the continuation of the citric acid cycle, helping to generate additional ATP and other important metabolic intermediates. And lastly, pantothenic acid. Pantothenic acid or vitamin B5 is a precursor for coenzyme A or CoA, a vital coenzyme in cellular metabolism. CoA is central to many biochemical pathways, primarily those involved in energy production and nutrient metabolism. Coenzyme A and its vital roles. First is synthesis of acetyl-CoA. Its role: CoA is essential for the synthesis of acetyl-CoA, a key molecule in metabolism. Acetyl-CoA is produced by the combination of CoA and an acetyl group, usually derived from carbohydrates, fats, or proteins. Its significance: acetyl-CoA is an entry molecule for the citric acid cycle or Krebs cycle. Once formed, it combines with oxaloacetate to form citrate, initiating the cycle that produces ATP, NADH, and FADH2, which are used for energy production. Without acetyl-CoA, the citric acid cycle cannot proceed efficiently. Next is the entry molecule for the citric acid cycle. Acetic CoA, derived from carbohydrates, fatty acids, and some amino acids, is a crucial entry molecule for the citric acid cycle. It enters the cycle by combining with oxaloacetate to form citrate, which undergoes further chemical transformations to produce energy-rich molecules. Its significance: acetyl-CoA's role in the citric acid cycle is fundamental to aerobic respiration, the process through which the body generates ATP from nutrients. This cycle is central to cellular energy production and metabolism. Next is fatty acid metabolism. Its role is that CoA is essential for the synthesis and breakdown of fatty acids. In fatty acid metabolism, CoA is involved in the activation of fatty acids before they can be transported into mitochondria for beta oxidation, where fatty acids are broken down to produce acetyl-CoA. Its significance: this process is crucial for energy production, particularly during fasting or prolonged exercise, when fatty acids become the primary energy source. Additionally, acetyl-CoA produced from fatty acids is used to generate ATP through the citric acid cycle. Lastly, amino acid metabolism. Its role is that CoA is also involved in the metabolism of certain amino acids. Some amino acids, when degraded, are converted into intermediates that can be converted into acetyl-CoA or other key metabolites that enter the citric acid cycle. This allows the body to derive energy from amino acids when carbohydrate and fat stores are insufficient. The ability to convert amino acids into intermediates that feed into the citric acid cycle supports the body's metabolic flexibility. The citric acid cycle, or CAC, and electron transport chain, or ETC, work together to fuel ATP production in cells. The CAC, which occurs in the mitochondrial matrix, oxidizes acetyl-CoA to produce NADH, FADH2, and ATP or GTP while releasing carbon dioxide. These high-energy electron carriers, NADH and FADH2, then enter the ETC in the inner mitochondrial membrane where they donate electrons, leading to the creation of an electrochemical proton gradient. This gradient powers ATP synthase, an enzyme that synthesizes ATP from ADP and inorganic phosphate. Oxygen acts as the final electron acceptor in the ETC, combining with electrons to form water. Together, the CAC and ETC are essential for producing the majority of ATP in aerobic respiration, with each NADH generating approximately 2.5 ATP and FADH2 producing about 1.5 ATP. Enzyme cofactors such as NAD+, FAD, and CoA play critical roles in these processes, ensuring efficient energy production for cellular functions. Why B vitamins matter? B vitamins are essential for efficient metabolism as they serve as crucial cofactors in various biochemical reactions, particularly those involved in the breakdown of carbohydrates, fats, and proteins. Without these vitamins, the body cannot effectively convert these macronutrients into usable energy. For instance, key enzymes in the CAC and ETC require B vitamin-derived coenzymes like NAD+, FAD, and CoA to function. Without adequate B vitamins, acetyl-CoA production is disrupted, halting the CAC and ETC, which are responsible for generating ATP, the body's primary energy source. This disruption leads to energy deficits and can impair numerous cellular processes, highlighting the critical role B vitamins play in maintaining metabolic efficiency and overall health.